Genetic diversity. What is genetic diversity? Well, you have high genetic diversity when there are many variants of a certain trait. An example of a large amount of genetic diversity inherent in a species is the variety of dog breeds that have been produced from the wolf genome. By crossing different offspring and then selecting for certain traits, like herding, retrieving, or dogs that have superior senses of smell, we end up cultivating a large amount of variation as shown by the different dog breeds. Think about it. The variety of dog breeds we have today actually reflects the extent of genetic diversity that was present in ancestral wolves, because the roots of that variation came from that wolf DNA. So let's talk about this in genetic terms. First, I want to review a few important terms. A locus. A locus is the site on a chromosome. It could be here, or here, or here. A locus is just the location, and it might be a gene that actually codes for a protein, or it might not be a gene. It could be a promoter region or something else instead. So locus just means a specific site on the DNA. An allele is a different variant of nucleotides at that locus on homologous chromosomes. So here we have 4 individuals with homologous purple chromosomes, and when we look at the same locus in each individual, as noted by the black arrow, we have different colors that represent the different variations of the DNA. These are called alleles. And you can see here that 2 of the individuals have the same color, meaning that they have the same allele. So in genetic terms, we have high genetic diversity when we have many alleles for a certain gene. If we think back to a gel, high genetic diversity is when we have many different sized bands. So if we remember the gel, the first column is the ladder, which shows us the standard sizes of base pairs for DNA. The 2, 3, 4, and 5 columns represent different samples. And then the products that are really short in base pair length move quickly through the gel and farther, and the products that are larger in terms of base pair size move shorter through the gel. So when we look at these and we try to figure out which ones are alleles, it's all of the bands that have the same number of base pairs. So here I've color-coded them. So the 2 blue ones are both the same allele. Then we have purple ones that are the same allele, a red one, and then a green one. So this shows us that we actually have 4 alleles in this gel. So why is having high genetic diversity important? Because of 2 reasons. 1, genetic diversity is required for populations to adapt to environmental change. And 2, the loss of genetic diversity is usually associated with inbreeding and overall reduction in reproduction and survival. So starting at the level of DNA, genetic diversity is different DNA sequences. These variations then code for different amino acids and then result in different proteins that are slightly different. The protein variation may result in biochemical, morphological, or behavioral differences among individuals that then cause differences in behavior, reproductive rate, or survival. That is why genetic diversity is important.